now make our way inside the cell and check out some of the internal structures. Now, eukaryotic cells contain a lot of membrane-bound organelles that we did not see in prokaryotic cells, as well as some cytoskeletal element. You'll see that they're vaguely grouped into components of the nucleus, into the membrane-bound organelles, ribosomes, and then the cytoskeletal elements. Let's begin by looking at the nucleus. The nucleus is separated from the cytoplasm by a nuclear envelope, which is a double lipid bilayer. So not just a bilayer, but two layers of lipid bilayer. As you can see illustrated over here, here's the outer lipid bilayer and the inner lipid bilayer. Now, these two lipid bilayers are perforated by nuclear pores that you can see just here. These nuclear pores allow traffic into and out of the nucleus. There's a region in the middle of the nucleus where we'll see RNA being transcribed from the DNA template. This is the region called the nucleolus. Most of the genetic material of the cell is contained within this double lipid bilayer nuclear envelope. In this micrograph down below, again, you can see a dark staining area that we consider to be the nucleolus inside this double lipid bilayer of the nuclear envelope. Note that the DNA is partitioned from the cytoplasm and the ribosomes, as opposed to what we saw in the prokaryotic cells, where there is no partition and DNA is in direct connection with the ribosomes inside the cytoplasm of the prokaryotic cell. The genetic material that we see inside the eukaryotic nucleus consists of multiple linear chromosomes, as opposed to the single circular chromosome that we saw in prokaryotic cells. Now, the eukaryotic cell can be either haploid or diploid. The natural adult stage, most commonly, is diploid, and most often we'll see that the gametes are haploid. The DNA that we find in the nucleus during most of the cell's life cycle is highly condensed. It's wrapped around histone proteins, so it's not really accessible for transcription and translation. But just prior to cell division, we're able to replicate this DNA by mitosis or meiosis, at which point it unwraps completely and the cellular mechanisms will replicate the DNA. You'll recall most of these cell structures and those processes from your general biology class, so consider that this is just a review of eukaryotic cell structures. Let's take a quick look to review how the cell cycle works.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into three discrete phases called G1, S, and G2. During the G1, or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing 2 copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has 4 stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attach to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the 2 groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, The chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of two genetically identical cells. To summarize the entire cycle, G1, S, and G2 phases occur during interphase while the cell is growing and preparing to divide. Mitosis, or the division of the nucleus, consists of four phases: prophase, metaphase, anaphase, and telophase, and in these phases duplicate copies of the chromosomes are split equally. Finally, the cell's cytoplasm divides during cytokinesis and results in the formation of two genetically identical daughter cells.
So this figure gives us a great review of the cell cycle, but do keep in mind this interphase is composed of several other phases, G1 and G2 and the synthesis phase, before we actually go into prophase. So the cell is actually in interphase for the majority of the cell's life. We only go into meiosis or mitosis, where we see prophase, metaphase, anaphase, telophase, etc., when the cell's ready to undergo division. Now let's look at some more organelles. We'll look into the endomembrane system within the cell that begins with the endoplasmic reticulum. The endoplasmic reticulum is a phospholipid bilayer. There's both smooth endoplasmic reticulum and rough endoplasmic reticulum. The difference here is simply that there are ribosomes docked on the rough endoplasmic reticulum. Otherwise, it's structurally the same, made of phospholipid bilayer, which is very similar to the cell membrane itself. In this figure, you can see the ribosomes studding the endoplasmic reticulum. So that's rough endoplasmic reticulum. And because it's got ribosomes all over it, it's involved in protein synthesis, specifically synthesis of proteins that will be exported from the cell. Now, the smooth endoplasmic reticulum is involved in lipid metabolism and detoxification reactions. So, for example, cells of the liver have an immense amount of smooth endoplasmic reticulum, because the liver is the organ that's involved in most detoxification. Now let's take a look at eukaryotic ribosomes. As we alluded in the prokaryotic section, eukaryotic ribosomes have a different molecular structure. They still have two subunits, a 40S subunit and a 60S subunit, and the total size becomes 80S, rather than the 70S that we saw in prokaryotic ribosomes. Ribosomes are used to translate the messenger RNA that comes from DNA in the nucleus into the proteins. You'll remember from general biology, we talked about transcription and translation, or the expression of genes into proteins. Now, these eukaryotic ribosomes have a very similar structure to those of prokaryotes, but a little bit different of a shape, and that contributes to their different weight. Also to note, eukaryotic ribosomes are antibiotic resistant, so we can't use the antibiotics that attack ribosomes to kill eukaryotic cells, which is what makes some of our antibiotics so valuable in treating microbial infections. Next in the endomembrane system is the Golgi apparatus. Now, once proteins are manufactured in the rough endoplasmic reticulum, they need to be transported out of the cell. The Golgi apparatus is like the shipping center, or packaging and shipping center. Here you take vesicles that have come off of the endoplasmic reticulum, and they travel to the arrival side, or the cis base, of the Golgi apparatus. They fuse with the phospholipid bilayer membrane there, and then they are shipped out again from the other side, repackaged and labeled for their destination, where they may or may not fuse with the cell membrane and make their way outside of the cell. So the Golgi apparatus is involved in trafficking protein. Again, it's composed of a lipid bilayer, and it's involved in import and processing and export. It looks very much like a stack of pancakes. While we're here, let's take a quick concept check. What is the nature of a typical eukaryotic genome? Is it a single linear DNA molecule? A single circular DNA molecule? Multiple linear or multiple circular DNA molecules? Well, you'll recall that prokaryotes had a single circular one, so we can cross that off our list. And We know that eukaryotic chromosomes are linear, so we'll keep these two linear ones on. Now the question is, do we have multiple or a single linear? Well, as you probably already know, we have multiple linear DNA molecules, 23 pairs, totaling 46 chromosomes. So we've covered the nucleus, and we've covered the endomembrane system, consisting of the endoplasmic reticulum and the Golgi apparatus. Now let's look at the double membrane-bound organelles. You'll recall there are two, the mitochondria as well as the chloroplasts. Now the mitochondria are the powerhouse of the cell. All cells have mitochondria because they need to generate ATP in order to do their daily functions. As we covered earlier, both mitochondria and chloroplasts most likely arose by the process of endosymbiosis, where a larger prokaryotic type cell engulfed some smaller prokaryotic type cells, and this is how they became double membrane bound. You can see this double phospholipid bilayer here. We have the outer membrane surrounding the entire cell, and then a convoluted inner mitochondrial membrane. Recall that mitochondria have their own genetic material and they replicate on their own. So that means they don't have to wait for the cell to divide in order to produce more mitochondria. Now, some cells have a lot of mitochondria, those cells that have a high demand for ATP, while other cells have many less mitochondria because they simply have less demand. Now let's take a look at chloroplasts. Chloroplasts are found only in plants and photosynthetic algae, because they are the site of photosynthesis. Again, they're a bacterial endosymbiont. That is, they probably originally came about because a larger prokaryotic cell engulfed a smaller one that was capable of photosynthesis. Just as with mitochondria, we see a double membrane-bound organelle. So here is our outer membrane, and then the inner membrane is also highly convoluted and forms these thylakoid stacks or grana. So these are just convolutions of the inner mitochondrial membrane. Also, like mitochondria, chloroplasts contain their own genetic material and can divide autonomously. Next, we move on to the cytoskeleton. This is an intracellular protein matrix, much like our own skeleton. It helps regulate the shape of the cell, as well as being involved in motility. We'll see three general types of cytoskeletal elements: the microtubules, the intermediate filaments, and the microfilaments. Now, actin is a type of microfilament, and you can see here that it's polymerized into chains. You also may recall from anatomy and physiology that actin is one of the primary fibers in muscle contraction. So muscle cells have lots of actin fibers. You can also see that there are microtubules here. Microtubules often provide highway upon which the cell can transport things, as well as some intermediate filaments here that are branched. Many cells also produce vacuoles. These are lipid-enclosed places, so they're essentially like a vesicle, but much larger. Vacuoles can be used for storage of food and water, as well as enzymes and waste products before disposal from the cell. They vary greatly in size and function. For example, in a plant, a central vacuole is filled with water and provides turgor pressure. So as the central vacuole becomes full, the cell becomes more turgid or more tight inside the cell wall. In many protozoans, we'll see contractile vacuoles. And contractile vacuoles are used to consistently eliminate water from within the cell. So water diffuses into the cell constantly because of the osmotic balance. So the cell doesn't burst. A contractile vacuole will suck in some of that water and literally squirt it out to the outside of the cell. Lysosomes are also a specialized type of vacuole. Sometimes lysosomes contain digestive enzymes. You can see in the figure on the right here the role of a lysosome in digesting food vacuoles. So we have some food here that has been engulfed by endocytosis, and now there's a food vacuole inside the cell. When it combines with a specific type of lysosome called the phagosome has provided enzymes necessary to digest the food. Digestion occurs, and the nutrients can then be released. Let's take a look at how those work by looking at this next animation.
Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. The hydrolytic enzymes degrade proteins, nucleic acids, lipids, and carbohydrates and are formed in the endoplasmic reticulum. These enzymes are then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles such as old mitochondria. This results in the destruction and recycling of these structures.
So let's have a quick concept check before we wrap up this section. Some eukaryotic cells do not perform aerobic respiration. So which cellular organelle can these cells do without? Now, all eukaryotic cells certainly need a nucleus, so we can cross that one off the list. What was the Golgi body involved in, or the Golgi apparatus? That was transport out of the cell, so that doesn't have anything to do with aerobic respiration. Ribosomes are about protein synthesis, so what we're left with is mitochondria, the powerhouse of the cell. They undergo aerobic respiration in order to produce ATP. Some other eukaryotic cells don't perform aerobic respiration to produce their ATP. They have other mechanisms that we'll get into shortly.